This biology lesson is on Standard Bio 1.LS 1.2, focusing on the impact of organic macromolecules on cell functioning. This lesson is presented by advisors Dexter Flanagan and Kimberly Spates. Our do now today is a review of what you learned in your previous lesson on the different types of cells found in organisms and how the role of each cell type relates to its composition and the prevalence of different organelles within that cell. You will need pencil and paper for it. As you can see, there are 4 pictures of cells: a sperm cell, a nerve cell, a red blood cell, and a bacterium cell. Bacterium is the singular version of the word bacteria. 1 bacterium and 2 or more bacteria. Your task has 2 parts. The first part is easy peasy. Match the cell type to its function. Read the functions listed to the side. A, carry oxygen to different parts of the cell. B, send signals from the body organ to the brain and back to the body organ for response. C, swim through the female vaginal canal to fertilize the egg with genetic information from the male. And D, quick reproduction without variation. The second part takes a little thinking. Reflect on the cell structures in the picture. How does the structure help the cell to perform its function? Remember, you have pencil and paper. Jot down your responses. You have 5 minutes. You have 2 minutes left. Take a moment and review your responses and take this last minute to wrap it up. Okay, guys, hopefully you were able to match the cell type to its primary purpose and then identify how each cell structure helps it to perform its function. Take a look at your responses and let's compare it to some of these possible responses. We know that the red blood cells carries oxygen to different parts of the cell. We also know that the red blood cell does not have a nucleus. The nucleus was removed, therefore leaving it with a donut-like shape, allowing it to carry the oxygen. We know that the nerve cell sends signals from the body organ to the brain and back to the body organ for response. That allows it to connect to all of the other nerve cells. It does not reproduce because it does not have centrioles. The sperm cell is what assists in fertilizing the egg with the genetic information. Its body shape is made especially for that. And lastly, the bacterial cell does quick reproduction without variation. It does not have a nucleus, and that allows for the DNA just to float in the center of the cell. Easy peasy reproduction and simple separation. Take a picture of the chart if you like to help you with your notes. Our phenomenon for today. The My Plate plan is a personalized food plan based on your age, sex, height, weight, and physical activity level. It helps you figure out how many calories you need each day, what nutrients you need, and shows you food group targets, what and how much to eat within your calorie allowance. You can read the food labels of items to help you to determine what foods you should select to eat depending on what nutrients your body needs. Take a moment to look at what your plate should have when you eat. Does your plate usually look like this? Do you have more vegetables than meat on your plate? Do you have fruit and vegetables? Do you also have a dairy, such as yogurt or milk? Maybe almond milk or cashew milk? Use your paper and pencil to develop 2 questions you would ask a nutritionist about the nutrients your body needs to help you select the best foods to eat. I think that I would ask, why are these amounts recommended for me to eat? What would you want to know? You have 3 minutes to develop your 2 questions. Remember, you're developing 2 questions you may want to ask a nutritionist about the nutrients your body needs to help you select the best foods to eat. You have 1 more minute. As you wrap up your questions, you will keep your questions near you as we go through the next 2 lessons. Have your questions handy. We'll refer back to them shortly. The My Plate plan asks us to learn to read our food labels. Each label gives us the calories, cholesterol, sodium, carbohydrates. It even breaks carbohydrates down to show whether or not the carbohydrates contain fiber or sugar. Also, the label identifies the amount of protein found in the food item. The My Plate plan lists grains as part of your food choices. Grains include breads, pastas, and cereals. Let's look at the food label for breads and pastas. According to the food labels, what are breads and pastas mostly made of? Let's take 2 minutes. Remember, you have white bread, whole wheat bread, and organic pasta food labels. Be sure to look at all 3. 1 more minute. All right, let's see what you came up with. Absolutely. Carbohydrates. The white bread has 16 grams of carbohydrates. Whole wheat bread has 18 grams of carbohydrates. And organic pasta, drum roll, has 41 grams of carbohydrates. Did you know that cereal also fits in this category? That's right. Therefore, cereal like oatmeal, Raisin Bran, Fruit Loops, etc., will all be considered carbohydrates. Let's look at fruits and vegetables. According to the nutritional food labels, what are fruits and vegetables mostly made of? Here we have an apple and celery. Let's look at their food labels. You have 2 minutes. Okay, what did we come up with? Absolutely. Carbohydrates. This apple has 25.1 grams of carbohydrates, and celery has 1.2 grams of carbohydrates. This would be the same for most fruits and vegetables, such as strawberries, grapes, blueberries, bananas, spinach, broccoli, and more. All right, let's look at the meats. As for meats, pepperoni, deli sliced lunch meats, hamburger meat, steak, ribs, and more. Or what do you think that these are made of? You have your 2 minutes. All right, guys. What's the answer? Yes, protein. The turkey pepperoni is 9 grams of protein, and the ground round for a great hamburger is 21 grams. This would also be the case of other meats such as eggs. You may find that meats also have a high number of grams of fat as well. Meat is taken from an animal's body. Animals like us have muscles and fats. The meat is the muscle of the animal, and the fat is the fat attached to the animal's muscle. Lastly, here are the oils and cheeses. According to the nutritional food labels, What are oils mostly made of? And secondly, what do we notice about the cheese? Let's take our 2 minutes. What did we find? What did we find? Yes, oils are made up of fats. Olive oil has 14 grams of fat. Other oils may be cooking oil, coconut oil, sunflower oil, and even butter. But what did we notice about that cheese? We noticed that cheeses, because they're made from milk, have both high levels of fat and protein. This cheese has 6 grams of fat, and it also has 7 grains of protein. Let's do some checking for understanding. Let's recall what 3 nutrient categories did all of the food items fit? I know you guys know. Let's see. Absolutely. Carbohydrate, protein, and fat. And this is what we're going to be looking at today. Remember your 2 questions that you developed from the beginning of the phenomenon? We asked that you kept them near. We're going to call them investigative wanderings. Now that you've learned a little about the 3 main categories of nutrients that your body needs, review your investigative wanderings and see if you need to adjust them. You're going to revisit your original 2 questions, which we're calling investigative wanderings. You're going to consider what you've learned about the 3 food-based nutrients needed for your body, and you're going to revise your questions if you see it necessary. You have 2 minutes. One more minute to wrap up your revisions. As you finish your questions, we want to remind you that you're going to keep your revised questions near you throughout the next 2 lessons. Today's essential question: what is the structure and function of the macronutrients necessary for our bodies to survive? I wonder why they're called macronutrients. Macro does mean large, micro means small. In the simplest form, macronutrients are the elements in food needed for a person to grow and function. They are needed in large quantities in comparison to other nutrients, which is why they are called macronutrients and are commonly referred to as macros. When looking at their purpose in the body, they are called macromolecules or biological macromolecules, or even for short, biomolecules. Lots of names, huh? Today's state standard and learning performance. The state standard for today's lesson is Bio 1.LS 1.2. Evaluate comparative models of various cell types with a focus on organic molecules that make up cellular structures. Our learning performance is: the students will obtain, evaluate, and communicate information in order to show the structure and function of the macromolecules that are necessary for survival, highlighting that structure determines many properties and functions of cellular organelles. For three-dimensional instruction, our science and engineering practice, or SEP, is obtain and evaluate and communicate information. Our disciplinary core idea, or DCI, is organic macromolecules. And our cross-cutting concepts, or CCCs, is structure and function. Our task for today's lesson: we will be implementing science and engineering practice, obtaining, evaluating, and communicating information. Our task includes watching a series of short videos, taking Cornell notes with predefined look-fors, and answering probing questions that identify the structure and function of the 3 macronutrients found in the body. Are you familiar with Cornell notes? Cornell notes allow you to write your look-fors on the left of your paper and your notes on the right. We will use Cornell notes to seek out the information on the structure and function of each macronutrient. Video number 1. What are carbohydrates? Take 2 minutes with your pencil and paper to copy your look-fors on the left side of your paper for your Cornell notes. Afterwards, we will begin the video. Let's take 2 minutes to jot down our key look-for information. Number 1, what elements do carbohydrates contain? What does a carbohydrate look like? Draw it. How does a monosaccharide make a disaccharide? What is the difference between a structure that ends in OSE and a structure that ends in ASE? What is the enzyme that digests sucrose? What are some examples of carbohydrates? How is a carbohydrate used in the body? Take 1 more minute to jot down your look-fors, and then we will start the video.
Plants use light energy from the sun to form glucose from carbon dioxide and water, according to the equation below. This process is photosynthesis. When we eat plant matter, we break down glucose in a reverse process called respiration. So what is so special about glucose? In this lesson, we will learn a bit more about glucose and a class of molecules that can be respired to release energy called carbohydrates. These carbohydrates, as suggested by its name, contain carbon, hydrogen, and oxygen atoms. It was previously thought that all carbohydrates have this generic formula, whereby there are 2 hydrogen atoms and 1 oxygen atom for every carbon atom. But we know now that some larger carbohydrates do not follow this generic formula, so it is no longer used. Glucose is a monosaccharide, or a simple sugar made of 1 subunit, as the prefix mono suggests. Disaccharides are made of 2 monosaccharides joined together by a glycosidic linkage. The 2 monosaccharides will undergo a condensation reaction to form the disaccharide. A condensation reaction is one where 2 molecules combine to form a larger molecule via the loss of a smaller molecule, in this case, water. Table sugar is actually sucrose, which is a disaccharide formed from fructose and glucose. Lactose, a sugar found in milk, is also a disaccharide formed from galactose and glucose. Have you noticed that all the simple sugars that we've discussed contain the suffix -ose? The enzymes responsible for breaking down the glycosidic linkage in the disaccharides have the same prefix as the disaccharide and with the suffix -ase. People who are lactose intolerant do not have sufficient lactase in their gut. Lactase is the enzyme that digests lactose. What is the enzyme that digests sucrose? Pause, think, and resume when ready. The answer is sucrase. When you eat starchy foods, such as rice, bread, potatoes, and pasta, you are actually consuming glucose. You may, however, wonder why they don't taste sweet. This is because the glucose monomers are joined together to form a polymer chain. This is like beads on a necklace. The individual beads are the glucose subunits, and the entire necklace is the polysaccharide molecule. Starch is actually a combination of 2 polysaccharides, amylose and amylopectin. The amylose is just like the necklace. It is a long chain of glucose monomers coiled into a helical structure. The amylopectin is like amylose, but with branches of glucose chains. The next time you eat a spoonful of rice, a piece of bread, or a forkful of pasta, try to chew it for 2 minutes without swallowing. You will find that it actually starts to taste sweet. There is an enzyme in your saliva called amylase. Here is a challenge. What do you think its function is? Pause, think, and resume when ready. The answer is amylase breaks down amylose into simple sugars. Now, we don't actually store glucose as amylose or amylopectin. This is how plants store glucose. The starchy foods that we consume are broken down into individual glucose subunits. Think of it as removing beads from the necklace. The glucose is used to build a polymer called glycogen, which is our main long-term energy storage molecule. It resembles a myelopectin, but it is more highly branched. When we need energy, glucose subunits are broken off from glycogen and subjected to respiration, thereby releasing energy. People who have diabetes either have an insufficient amount of the hormone insulin, which signals the liver and muscle to link glucose into glycogen, or those liver and muscle cells do not respond to the presence of insulin. Cellulose is another polysaccharide made of glucose monomers. It has a more rigid structure and is the main component of plant cell walls. Your paper is actually made of cellulose. If you tear off the corner of your paper, you will see that there are fine hairs on the edge. These are cellulose fibers. In summary, these carbohydrates are polymers of simple sugars, made of carbon, hydrogen, and oxygen atoms, which provide fuel for living organisms. The energy is transferred when the sugars react with oxygen during respiration. They can exist as monosaccharides, disaccharides, or polysaccharides. Humans store fuel in the long term in the form of glycogen, which is a polysaccharide made of glucose subunits. This can be broken back into simple sugars, which can be respired when required to give us energy.
All right, now that we've seen our video on carbohydrates, let's take a moment to review the look-fors for the structure and function of carbohydrates that we've taken with our Cornell notes. Ensure that you have responded to each of these questions. You have 2 minutes to clean up your notes and ensure that you have a response for each of the checks for understanding questions. Okay, let's look at our possible answers. Here are the possible answers for the look-fors on the structure and function of carbohydrates. Pay close attention to the illustrations. Carbohydrates contain carbon, hydrogen, and oxygen. A monosaccharide makes a disaccharide through dehydration synthesis. The difference between OSE and a structure that ends in ASE is a sugar ends in OSE, and the enzyme that breaks it down ends in ASE. The enzyme that digests sucrose is sucrase. And some examples of carbohydrates are pastas, breads, and rice. Carbohydrates are used in the body for energy release, and it provides ongoing energy within the body. What are lipids? Video number 2. Take a moment, about 2 minutes, to copy down your look-fors on the left side of your paper for your Cornell notes, which you will take on the right side of your paper. After these 2 minutes, we will begin the video. Remember, during the video, you're looking for the answers to these particular look-fors.
Lipids are a group of hydrophobic biomolecules that play important roles in living organisms. While the primary function of lipids is long-term energy storage, lipids are also used for protection, insulation, and lubrication. They also act as precursors for some hormones and are a key component of cell membranes. There are 4 basic groups of lipids. These are triglycerides, phospholipids, steroids, and waxes. Although these groups differ in many respects, they all have one characteristic in common. They are all insoluble in water. You may have noticed that lipids and water do not mix. For example, notice the yellow-colored oil in the beaker of water shown here? Even if we stirred this for several minutes, or even several hours, the oil would still separate out from the water. This is because lipids are hydrophobic. From Latin, the prefix hydro means water, and phobic means fear of. So when you hear that lipids are hydrophobic, this basically means that water and lipids do not mix. Let's take a closer look at the category of lipids known as triglycerides. Triglycerides include the fats and oils. Fats,
such as lard and butter, are solid at room temperature and are used by animals for insulation, protection, and long-term energy storage. Oils, such as corn oil and olive oil, are liquid at room temperature and are used by plants for long-term energy storage. At the molecular level, triglycerides contain two types of subunit molecules, glycerol and fatty acids. Let's take a quick look at fatty acids. A fatty acid has three main parts: a chain of carbon and hydrogen atoms called the hydrocarbon chain, a methyl group at one end, and an acid group at the other end. Fatty acids can either be saturated or unsaturated. A fatty acid that has only single carbon-to-carbon bonds is known as a saturated fatty acid. This is because the carbon chain is saturated with all the hydrogen atoms it can hold. Unsaturated fatty acids have one to several double bonds. Double bonds result in kinks in the fatty acid chain, which affects the melting point of the fat. Animal fats have saturated fatty acids and are solid at room temperature, while vegetable oils have one or more double bonds and are liquid at room temperature. A trans fat is an example of an unsaturated fatty acid where the hydrogen atoms are on opposite sides of the double bond. Trans fats are usually formed during the production of processed foods and are also common in partially hydrogenated oils. In order to increase shelf life and melting point of the fat, excess hydrogen atoms are introduced to an unsaturated oil. This causes the formation of trans fat bonds in the fatty acid chain. Unfortunately, the consumption of trans fats has been associated with cardiovascular disease, and its use has fallen from favor. Now that you understand a little bit about fatty acids, let's zoom back out and look at how the triglyceride subunits fit together. Remember, a fatty acid is only a small part of a triglyceride. To become a triglyceride, three separate fatty acids have to bond with a glycerol molecule through the process of dehydration synthesis. Let's move on to the next category of lipids, which is phospholipids. Phospholipids are similar to triglycerides in that they contain glycerol and two fatty acids. What's different is that a phosphate group rather than a third fatty acid is attached to the third carbon of glycerol. Phospholipids are extremely important, mainly because of their unique properties in regard to water. The phosphate head of the molecule is hydrophilic, or water-loving. This means that it mixes well with water. The fatty acid tails, however, are hydrophobic, or water-hating, and do not mix well with water. Because of these unique properties, phospholipids tend to arrange themselves so that only the hydrophilic heads interact with the watery environment, and the hydrophobic tails crowd inward away from the water. This structure is the major component of plasma membranes of the cell. Steroids are the next category of lipids. Steroids are composed of four fused rings of carbon to which different functional groups are attached. One well-known steroid molecule is cholesterol. Cholesterol serves as a precursor for the synthesis of other steroids such as testosterone, estrogen, vitamin D, and cortisone. Cholesterol is present in plasma membranes where it stabilizes the membrane. The hormones testosterone and estrogen have small differences in their functional groups, but large differences in their effects on an organism. Waxes are the final group of lipids. Waxes are nonpolar and repel water. They're found in the protective coating on leaves and on outer surfaces of animals. Wax is produced in the ears of some animals to protect the eardrum. In addition, bees construct honeycombs from wax. Now that we've covered all four categories of lipids, let's do a quick recap. The four categories of lipids are triglycerides, phospholipids, steroids, and waxes. All lipids are insoluble in water. While the primary function of lipids is long-term energy storage, lipids are also used for a multitude of other purposes, such as protection and insulation, and as a key component of hormones and cell membranes.
All right. Now that we have finished our video on lipids, take a moment to review the look-fors for the structure and function of lipids. Ensure that you have responded to each question. You have found the function of a lipid, the basic groups of a lipid, what all lipids have in common. You've identified the structure and an example of each of the 4 groups of lipids. You have 2 minutes to complete your notes and your responses for checking for understanding. Okay, let's look at some possible answers for your look-fors. Here are the possible answers for the look-fors on the structure and function for lipids. Pay close attention to the illustrations. You may want to take a picture for your notes. We know that lipids are long-term energy storage protection. They also are good for lubrication, hormones, the cell membrane structure, and insulation. The 4 groups of lipids are triglycerides, phospholipids, steroids, and waxes. All lipids are insoluble in water. Triglycerides are fats and oils. Phospholipids can be found in the cell membrane. Steroid examples are testosterone, estrogen, and vitamin D. And waxes, we know about earwax, honeycombs, and protective coatings on leaves. Please be sure to include your illustrations for the structure of each type of lipid. Our last video. What are proteins? Take 2 minutes to copy down your look-fors on the side of your paper, on the left side of your paper for your Cornell notes, because you will be taking notes on the right side. After that 2 minutes, we will begin the video. We want to ensure that we're able to find what proteins are made of, how many building blocks of proteins there are, what proteins look like, and we want to ensure that we have a drawing of it. We want to identify common examples of proteins. We want to know how a scientist would test for a protein. And lastly, we're looking for where proteins are used. Take this last minute to wrap up your questions on the left side of your paper for your Cornell notes. The video will follow shortly.
What do all of these things have in common? They are all made of protein. In this video, we're going to discuss the structure and uses of protein molecules. Proteins are long-chain molecules made of strings of amino acids joined together. All amino acids are compounds with a similar basic structure. They have an amino group, a carboxyl group, and a side chain, or R group, that is specific to each amino acid. There are 20 different amino acids. All of the different proteins are made from these 20 amino acids. The variation in proteins is caused by the order of amino acids, the chain length, and by folding the chain in distinct ways. As part of a balanced diet, roughly, how much of it do you think should be protein? About 14%. Sources of protein in our diets include meat, fish, eggs, cheese, beans, and nuts. But did you know that proteins are large molecules, too large for our bodies to absorb? That's why enzymes in our digestive system break down proteins into amino acids, which are small enough to pass from our digestive system into our blood to be assimilated by the body. When we absorb these amino acids, they are then built back up into proteins and used in a variety of ways. To test food for protein, you need to use copper sulfate solution and sodium hydroxide solution. These two liquids together may be called Biuret solution, which is a pale blue liquid. To begin the first experiment, first make a solution of the food sample by dissolving it in some water. You may need to grind this up first with a pestle and mortar. Next, add a few drops of sodium hydroxide solution to your food solution. Then a few drops of copper sulfate solution. If the solution turns lilac or purple, the food contains protein. If the food solution remains blue, no protein is present. But how does our body build proteins from amino acids? Our DNA acts as a set of instructions for how to build proteins. Each individual's DNA is made of slightly different genes, which explains some of the differences between people. For example, hair color and eye color. A change in the DNA, called a mutation, can lead to a protein with a different structure being produced. This can lead to albinism, for example. To learn more about mutations, you can watch this video. So what are proteins used for in the body? The answer is, they are used in thousands of ways. Our hair, nails, skin, and muscles are all examples of structures made of protein. Many hormones, including insulin, are made of protein. Antibodies, made by white blood cells to protect us from diseases, are also made of protein. Enzymes, biological catalysts found inside our body are made of proteins too. You can learn more about enzymes in this video. And last but not least, proteins are also used to move some substances across cell membranes in active transport. For example, to absorb glucose in the small intestine. So now you can see just how important
Thank you for paying attention to our last video, video number 3 on proteins. Take a moment to review the look-fors for the structure and function of a protein. Ensure that you have responded to each question. Take these 2 minutes to review your Cornell notes and to ensure that you have responses for each one of these questions. You should be wrapping up your responses now. All right, let's look at the possible answers for your look-fors. Here are the possible answers for the look-fors on structure and function of a protein. Pay close attention to the illustrations. You may want to take a picture for your notes. We know that proteins are made up of chains of amino acids. There are 20 amino acids that are building blocks for proteins. What does a protein look like? A chain of blocks linked together. List some common examples of proteins: meats, eggs, cheese, also nuts. How would a scientist test for a protein? Drop some borate solution on the substance. If it turns purple, it is a protein. If it remains blue, it is not a protein. What are proteins used for? Well, proteins are everywhere. Your hair, your nails, skin, your muscles, insulin, antibodies, enzymes. They also assist with your cell membranes by assisting in moving substances across the membrane. Proteins have a very large job within our bodies. Wrapping up today's lesson, our question of the day was, what is the structure and function of macronutrients necessary for our bodies to survive? Today we investigated Standard Bio 1.LS 1.2, focusing on the organic macromolecules. To lead our actions and guide our assessments, our learning performance was the students will obtain, evaluate, and communicate information in order to show the structure and function of the macromolecules that are necessary for survival, highlighting that structure determines many properties and functions of cellular organelles. We engaged in a phenomenon of food labels where we determine the main categories of the foods that we eat. I'm sure now that when we go to the grocery store, we will read the labels to maximize the nutritional value of the foods that we select and purchase. We also obtained information today by using our Cornell notes to answer the probing questions through 3 videos on lipids, proteins, and carbohydrates. We drew the structure and were able to list the functions of each of the 3 macromolecules. In our next lesson, we will look closer into the impact of these macromolecules on the functioning of cellular organelles and their roles within the organisms. We're going to leave you with the final task, a 3, 2, 1 ticket out the door. Pull out those revised questions that you developed at the beginning of our lesson, your investigative wanderings. You're going to write 3 things that you learned, 2 things that surprised you, and 1 question that you still have. Have a great day.